Hello everyone, this is Dr. Preeti Mehta from Department of Pharmaceutical Chemistry, Navasahyadri Institute of Pharmacy, Pune. First of all, I would like to thank the coordinators of IMCCRT conference for giving me an opportunity to present my work entitled Simultaneous Estimation of Montelukast sodium and Acebrophylline in bulk and tablet dosage form by reverse phase high performance liquid chromatography method. Now, as we all are aware that the pulmonary disease and particularly asthma has become a major health issue in developing countries. And many drugs are available in the market for the treatment of these pulmonary diseases like Montelukast sodium and Acebrophylline. These drugs are available in tablet form as an oral combination. Montelukast sodium is a leukotriene receptor antagonist and it is used to treat the asthma and to relieve symptoms of seasonal allergies. Acebrophylline, which is another drug, it is a xanthine derivative and it is used as a bronchodilator for the treatment of bronchial asthma and COPD in adults. As per literature review, there are few methods available for the estimation of Montelukast sodium and Acebrophylline individually in bulk and in pharmaceutical formulations. These are spectrophotometric, spectrofluorometric, RP-HPLC and HPTLC methods. But these methods are used to determine the concentration of these drugs in bulk and in pharmaceutical formulations individually. But very few analytical methods have been reported till date for the estimation of combination of these two drugs. So, the objective of the present research work is to develop a rapid, simple, precise and accurate reverse phase liquid chromatography method, that is RP-HPLC method, for simultaneous estimation of Montelukast sodium and Acebrophylline in combined dosage form and to validate this method as per ICH guidelines. The materials required for this work like drugs, pure drugs, Acebrophylline and Montelukast, they were supplied by Lupin Pharmaceutical and MacLeod's Pharmaceutical respectively. HPLC grade methanol was procured from Merck and Qualigen Fine Chemicals. The HPLC system consisted of a pump. Model is Waters 515 HPLC pump with auto sampler was used. The detector in HPLC consisted of a photodiode array which is operated at a wavelength of 245 nanometer, and the columns used for the study were Symmetry C-18 and kromasilC. Now moving to our methods, the first step was optimization of HPLC method, where the pure drugs, that is Montelukast and Acebrophylline, they were injected and run in different solvent systems. Initially, acetonitrile and water in different ratios were tried. In these solvents, both drugs showed development, but peaks were not sharp. That's why acetonitrile was replaced by methanol and different ratios again were tried. In this mobile phase, both the drugs showed development. That's why different ratios of methanol and phosphate buffer were then tried. Finally, the mobile phase containing methanol as for phosphate buffer in the ratio of 85 to 15 plus glacial acetic acid 5% was tried. Here, the pH of the mobile phase was adjusted in the range to 3.5 by adding glacial acetic acid. In this mobile phase, the symmetry and the resolution of standards were found satisfactory. That's why at the end, methanol, combination of methanol, phosphate buffer and 5% glacial acetic acid was selected as a mobile phase for the validation purpose. These are the chromatographic parameters selected. The column selected was Symmetry C-18, mobile phase, methanol as to mixture of methanol, phosphate buffer and 5% glacial acetic acid. Flow rate was 0.8 ml per minute, wavelength 245 nanometer and temperature 40 degrees centigrade. Next step is preparation of standard solutions and calibration graphs. For this, stock solution was prepared by dissolving 25 mg of standard Montelukast and standard Acebrophylline in 25 ml of mobile phase. The standard solutions were then further prepared by diluting this stock solution with the mobile phase to get a concentration range of 4 to 20 microgram per ml for Montelukast and 80 to 400 microgram per ml for acebrophylline respectively. Now, the triplicate 10 microliter injections were then injected 2 times for each concentration with autosampler and chromatograph under the conditions described above. Then, calibration curves were obtained by plotting peak areas against the corresponding concentrations of drugs. Next step was analysis of formulation, where the 20 tablets of Telukast A, which is a combination of these two drugs, were weighed to determine the average weight, and these tablets were then triturated to get a fine powder. Powder equivalent to 25 mg of Montelukast and 25 mg of acebrophylline was weighed and transferred to a 50 ml volumetric flask, which contained 35 ml mobile phase. Now this solution was sonicated for 40 minutes and then further diluted to 50 ml with a mobile phase. This sample solution was then centrifuged after sonication for 15 minutes. After centrifugation, the supernatant was separated, and after appropriate dilution, the sample solution was then filtered using 0.45 micron nylon membrane filter. Now the above stock solution was further diluted to get the sample solutions of different concentrations, and the 10 microliter volume of each of these sample solutions were then injected into HPLC 6 times under the specified conditions, and the peak areas were measured at the wavelength of 245 nanometer. The developed analytical method was then validated with respect to linearity, precision, accuracy, robustness, limit of detection and limit of quantitation. So first parameter which was validated was linearity. Here the 8 calibration standard solutions were prepared over the concentration range of 4 to 20 microgram per ml for Montelukast and 80 to 400 microgram per ml for acebrophylline. All these solutions were then injected into the chromatograph, and then peak responses for each level were recorded, and the parameters like slope, intercept, regression coefficient, correlation coefficient were calculated, and calibration curves of area against concentration were plotted for each concentration for both the drugs. Next parameter validated was precision, where the standard solution preparations were injected 6 times into the chromatographic system and peak responses were recorded. The repeatability of sample application and measurement of peak area for active compound were expressed in terms of % relative standard deviation. Third parameter was accuracy, which was evaluated by performing recovery studies. It was carried out by applying the HPLC method to drug content present in tablet dosage form, to which known amount of mixed standard of Montelukast and acebrophylline were added at 80%, 100% and 120% levels. Fourth parameter was limit of detection and limit of quantitation, which were calculated using the standard formula. Next parameter validated was robustness. So to determine the robustness of the proposed method, aliquots from homogeneous lots were analyzed by varying the physical parameters like mobile phase composition, flow rate, column temperature, pH of buffer and wavelength. Now these are the results and discussion part. So first, HPLC procedure was optimized with a view to develop a simultaneous assay method for Montelukast and acebrophylline. Table 1 indicates the system suitability parameters. So here the first parameter is retention time. For Montelukast, retention time was found to be 3.5. For acebrophylline is 4.3. Telling factor for Montelukast was 1 and acebrophylline 1.4. Theoretical plates. These are the theoretical plates 147340, 1,47,340, and for acebrophylline 5,52,220. Asymmetry values for Montelukast is 1.1 and acebrophylline 1.4. Now the results of method validation. First parameter is linearity and range. So it was found that the Now these are the calibration curves for Montelukast and calibration curve for acebrophylline, and these tables indicate the linearity data. So the linearity was determined for Montelukast in the range of 4 to 20 microgram per ml, and for acebrophylline 80 to 400 micrograms per ml. So here in the image you can see the linearity range is between 4 to 20 microgram per ml for Montelukast and between 80 to 400 microgram per ml for acebrophylline. Here are the values of regression coefficient equation over here. Next is precision. So the repeatability of the sample application and measurement of peak area for active compound were expressed in terms of percent relative standard deviation. These are the results of inter-day precision. For Montelukast, the repeatability of method was determined at the concentration of 12 microgram per ml, where the percent relative standard deviation was found to be 4.058. For acebrophylline, the precision was determined at the concentration of 240 microgram per ml, and the percent RSD was found to be 1.314. These are the peak areas for the Montelukast and acebrophylline. Third parameter, accuracy, which was determined by performing recovery studies at 3 different levels, 80, 100 and 120%. This is the accuracy data for Montelukast. So at 3 different accuracy levels, 80%, the amount of Montelukast in sample solution was found to be 10 microgram per ml, in standard solution 8 microgram per ml. So total amount of Montelukast was 18 microgram per ml, and by performing recovery study, total amount of Montelukast found was 18.26. So the percent mean recovery was 98.13. At 100% level, the percent mean recovery was found to be 99.5%, and at 120% accuracy level, the percent mean recovery was found to be 99.83%. This is the accuracy data of acebrophylline. Same, the accuracy level were 80%, 100% and 120%. At 80%, the percent mean recovery of acebrophylline was found to be 99.07. At 100% accuracy level, the percent mean recovery of acebrophylline was 101.06%, and at 120%, the mean recovery was 100.75%. So the proposed method was found to be accurate. Next parameter validated was limit of detection and limit of quantitation. So to determine the LOD and LOQ, a known concentration of each drug was diluted until the signal to noise ratios of approximately 3 and 10 were obtained respectively. This table shows the LOD and LOQ values. So the limit of detection for acebrophylline was 0.813 microgram per ml, for Montelukast 0.619 microgram per ml, while limit of quantitation for acebrophylline is 2.4 microgram per ml, and for Montelukast it is 1.8 microgram per ml. So these results indicate the capability of method to detect and quantify low concentrations of these two drugs. And the last parameter validated was robustness of the method. So to evaluate the robustness of method, a few parameters were changed, like flow rate, pH of buffer, column temperature, percentage of methanol in the mobile phase, and wavelength. So results of the same were displayed over here. So first variable was flow rate. The flow rate was changed from 0.6 to 1. So first one is 0.6 ml per minute. At that flow rate, retention time for acebrophylline is 4.6, for Montelukast 3.7. Telling factor was 1.2 and 1. For 0.8 ml per minute, the values were again similar, and for 1 ml per minute, the retention time and telling factor was found to be same. So though flow rate was changed, the results found to be close or similar. Next variable was pH of the buffer, where the pH was changed from 3.2, 3.5 and 3.7. So here again, retention time for both the drugs and telling factors for both the drugs, these values were found to be almost same. Third variable was column temperature. So the temperature of the column first was 38 degree, then it is changed to 40 and then 42. So at these variable temperatures, the retention time and the telling factors of both the drugs were found close to the standard values. Next variable is ratio of mobile phase. So here the ratio of mobile phase, the first ratio is 83:17. The retention time for acebrophylline was 4.5, Montelukast 3.7. Telling factor for acebrophylline 1.3 and Montelukast 1.1. Again the ratio is changed from 85 to 15. The retention time and telling factors for both the drugs were similar as that of the earlier. And the third ratio was 87:30. So the retention time and telling factors for both the drugs were again similar. So though the ratio of mobile phase was changed, the retention time and telling factor values found to be same or similar. Next variable was wavelength. Wavelength variables were 243 nanometer, 245 nanometer and 247 nanometer. Again at these variable wavelengths, the results were found same. So the conclusion of the present research work is a simple, sensitive, accurate and specific RP-HPLC method, that is reverse phase high performance liquid chromatography method, was developed for the determination of Montelukast and Acebrophylline in pure form and in pharmaceutical formulations. The developed method was validated according to ISAGE guidelines and it was found to be accurate, precise and robust. Thus, the proposed method can be successfully applied for simultaneous determination of Montelukast sodium and acebrophylline in bulk as well as in combined tablet dosage form. These are the few references for the present research work. So this is all about my presentation. Thank you so much.